Hello students, a very warm welcome to my YouTube channel. I'm Dr. Arpita De and I hope you all are doing great. The topic for today's discussion is Lac Operon. Lac Operon is studied to get an idea about the gene regulation procedure in prokaryotes. Before we get into the topic, let's first define an operon. An operon can be defined as a group of genes that are transcribed together as a single mRNA under the control of a single promoter. The basic structure of the operon is described here. Primarily, the operon consists of a regulator gene, a promoter gene, and an operator gene. The details of the structure and their functions will be discussed here. So now let's talk about the structure of Lac Operon. This is the entire structure of Lac Operon. Let's start with this. This one is I. This codes for the repressor molecule. This is the repressor. This repressor is constitutively transcribed. Now coming to this one. This part is the cap binding site. Now what is this? This is the promoter region. This is the operator. Now we are left with these three. These three are the structural genes. Now we will see what are these genes encoding. These three genes will be encoding for three enzymes. This is LAC-Z, this is LAC-Y, and this is LAC-A. Now let's see what are these three genes coding for. These three genes will make three different enzymes. LAC-Z will make beta galactosidase. What will this do? This will break down glucose. This will break down lactose into glucose and galactose. Glucose and galactose. Okay. Now we'll see what is LAC-Y making. LAC-Y gives us galactose permease. What is this function? It transports lactose into the cell. Okay. Transports lactose into the cell. Now finally we'll come to LAC-A. What is this doing? This is giving us thiogalactoside transacetylase. Thiogalactoside transacetylase. This normally helps to get rid of thiogalactoside. Now, this is the picture of the structural composition. Now, let's see what is this red ball doing. This is RNA polymerase. Now, lactose is when lactose is not there in the medium, what is happening then? This repressor molecule will go and bind to which one? This repressor molecule will go and bind to the operator region like this. When this is bound to the operator region, it gives a signal which we say off. This signal is a hindrance to the performance of this RNA polymerase. The RNA polymerase, as a result, cannot bind to the promoter. Therefore, there is no transcription. Transcription does not occur. On contrary, when this is lactose, when lactose is present in the medium, then what happens? Then transglycosylation by beta-galactosidase produces allolactose from lactose. Now this one will go and bind to the repressor molecule. When this binds to the repressor molecule, the repressor is no longer able to bind to the operator. This will give a signal which we say on. This on signal will enable the RNA polymerase to bind to the promoter. When RNA polymerase binds to the promoter, this allows transcription of structural genes. At that time, what will happen? These three structural genes will be transcribed, and as a result, what will be produced? Beta-galactosidase, galactose permease, and thiogalactoside transacetylase. These three enzymes will be produced in the presence of lactose. Now we will talk about the performance of Lac Operon in the presence or absence of different types of carbon sources in the media. The first case is talking about glucose and lactose, their presence and absence respectively. We will talk about the performance of Lac Operon when glucose is present in the media and lactose is absent. We all know glucose is a preferred source of carbon. Therefore, when glucose is there, it will be utilized. But here we see lactose is absent. So what will happen? The repressor molecule that is produced by LAC-I gene or the regulator gene will remain bound to the operator and thus prevent the binding of RNA polymerase to the promoter region. As a result, what will happen? The structural genes will not be transcribed. The second case here shows absence of both lactose as well as glucose. It's quite evident that if lactose and glucose both are absent, the operon will not perform, or else there will be no transcription of genes at all. The same condition applies here also. The repressor will remain bound to the operator region. The RNA polymerase will not bind to the promoter region, and hence there will be no transcription. So the result of the previous two cases is what? No transcription of structural genes. Now we will come to the third condition. The third condition is where glucose is present and lactose is also present. What will happen here? Here also we see no transcription. Why? Because RNA polymerase will be able to sit on the promoter, but the binding is not stable, and as a result what will happen? It will keep off falling. And if it falls off, there will be no transcription of the structural genes. The last condition is what? Glucose is absent and lactose is present. This is the condition where we see the structural genes are getting transcribed. The operator is no longer blocked by the repressor, since allolactose binds to it and keeps it from binding to the operator. When lactose is present and glucose is absent, then what happens? Cyclic AMP binds to the inactive cap and changes it to active cap that binds to the cap binding site. This allows the RNA polymerase to fully bind to the promoter. When this happens, it's evident that transcription will occur smoothly. So these are the four conditions when we see transcription is occurring and transcription is not occurring. So the last one is the only condition when we get to see the occurrence of transcription. Hope this video has helped you. If you find it interesting and helpful, do share it with your friends. We will be talking about the regulation and mutations in Lac Operon in the upcoming video. Thank you so much for your kind attention.